Usually when we think of a biological threat, we picture this perfectly calibrated machine, like a virus enters your body, and the assumption is that it has the exact right tools, the perfect genetic code to seamlessly hijack your cells and replicate.
Right, exactly. It's supposed to be this elegant, highly adapted, hostile takeover. I mean, evolution's a ruthless editor, so we naturally expect a successful pathogen to be fully optimized for its host environment.
But then you look at something like the COVID-19 pandemic. I mean, SARS-CoV-2 rapidly caused millions of deaths, operating with just terrifying efficiency inside human host cells. But mathematically and, well, biologically, according to the rules of translation we thought we understood, it shouldn't have been able to do that. It was using the completely wrong genetic dialect.
Yeah, it really is the absolute definition of a biological paradox.
Yeah.
It's the molecular equivalent of someone writing a sweeping, complex Shakespearean tragedy using only a suboptimal, archaic vocabulary, but somehow still publishing it faster than a modern printing press.
It's insane. Well, welcome to today's Deep Dive. I'm your host, and joining me is our resident structural biology expert. We are so thrilled you're joining us today.
Glad to be here.
So our mission today is to meticulously unpack a really groundbreaking 2025 Nature paper. This is by researchers Huan Liu, Yi Liu, and their team at the UT Southwestern Medical Center. And we are going to explore, step by step, how viral RNA uses structural geometry to evade the host cell's ultimate quality control mechanisms.
And we really need to emphasize right at the top that this isn't just, a niche virology story. This discovery fundamentally shakes up our common knowledge of how human cells control their own protein production.
Wow. Oh, yeah.
In fact, Nature editors Florian Ulrich and Martine Collard specifically noted that this paper is going to stir up significant controversy because it basically rewrites basic human cellular biology.
That's huge. So if you are at all interested in how viruses outsmart our immune systems, or if you want to understand the actual microscopic mechanics of how modern mRNA vaccines function, you absolutely have to understand the physical shape of these molecules.
Exactly. Shape is everything here.
But to get there, we first need to define the genetic language barrier that absolutely should have stopped these viruses in their tracks. So we already know viruses hijack host ribosomes, relying entirely on the host's transfer RNA, the tRNA pool, to build their proteins.
Right. Viruses are, they're the ultimate minimalists. They just don't have the genomic space to pack their own massive tRNA fleets. They have to use whatever's floating around in the human cell.
Makes sense.
And this brings us to the concept of codons and tRNAs. So imagine mRNA as a long instruction manual.
Okay, I'm picturing it.
Codons are the 3 nucleotide sequences, basically the 3-letter words in this manual. Transfer RNA, or tRNA molecules, well, they are the delivery trucks that read these words and drop off the corresponding building blocks, the amino acids, to build a protein.
Oh, I like that analogy. Delivery trucks. And that brings us to synonymous codons. So it's like how the words huge and gargantuan mean the exact same thing, right? You can use either one to describe a giant monster. In genetics, different codons can code for the exact same amino acid.
Precisely. But here's a catch. Human cells have a heavy codon usage bias. Even though huge and gargantuan mean the same thing, human biology heavily indexes on specific preferred codons. We call these the optimal codons.
Why is that?
Well, because the cell maintains a massive pool of the specific tRNA delivery trucks that are perfectly matched to read those optimal words.
Okay, let me make sure I'm wrapping my head around this. So if a transcript uses the optimal codons, the ribosome just flies down the mRNA strand. The correct delivery truck is always right there, ready to drop off its amino acid.
Exactly. It's a super efficient highway.
But if you use a non-optimal codon like the genetic equivalent of the rare word gargantuan, the cell only has a tiny fraction of delivery trucks available for that specific sequence.
Which causes a massive drop in translational efficiency. The whole cellular factory basically has to slow down and wait for that rare, non-optimal tRNA truck to randomly bounce into the ribosome.
Right.
Now, here is the paradox we teased at the start. The mRNAs of most human viruses, including SARS-CoV-2, are packed to the brim with these rare, non-optimal codons.
Okay, let's unpack this, because my initial thought when looking at this is, well, maybe the virus has some hidden workaround. Like maybe it forces the cell to produce more of those rare tRNAs during an infection.
That's exactly where a lot of early hypotheses went. Biologists just assumed there had to be some adaptation we couldn't measure yet. Another competing theory was that this core adaptation was actually, Well, intentional, a sort of biological truth.
They had truths.
Yeah, the idea was that the virus intentionally used non-optimal codons to slow down its own replication, ensuring it didn't kill the host cell too quickly so they could basically coexist.
Wow. But the reality on the ground during a severe infection totally contradicts that truth theory, right? I mean, viral protein expression in infected cells isn't a slow, measured trickle. It is incredibly efficient. It's at warp speed.
Exactly. The virus is using a suboptimal language, relying on a depleted delivery truck pool, and yet it is massively outproducing the host's own cellular machinery.
Okay, so let's do a quick recap of this first section so we don't lose anyone. Bullet number 1. Codons are 3-letter genetic words, and tRNAs are the delivery trucks.
Spot on.
Bullet 2. Host cells have a massive bias toward optimal words. And bullet 3. Viruses use non-optimal words, but mysteriously still build their proteins at warp speed.
That's the perfect summary.
So a quick question for you listening right now.
Ah.
Have you ever been in a situation where someone was using all the wrong tools but still managed to finish the job faster than the experts? How would you even begin to investigate that cheat code?
And that is exactly the methodological hurdle that Huanlu, Yilu, and their UT Southwestern team had to clear. Their laboratory detective work here is it's brilliant.
What did they do?
They realized you can't just look at the virus in a vacuum. You have to run a comparative experiment. They compared the translation outputs of viral mRNAs against host cellular mRNAs,
Okay.
specifically looking at sequences that contain both optimal and non-optimal codons. But the genius of their approach was in how they packaged these genetic sequences in the lab.
Right. They didn't just dump raw viral RNA into a dish. They tested the sequences in 2 very distinct formats.
Yeah, exactly. The first format was what we call human-like transcripts. They took the viral genetic code and artificially attached 2 very specific structural hallmarks of human mRNA. They added a molecular cap on the 5' end of the RNA strand, and they added a polyadenine tail, a poly-A tail on the 3' end.
So they essentially disguised the virus. They took the raw viral code and slapped the standard human front cover and back cover onto it, ensuring the host cell would recognize it as one of its own.
Precisely. And they even expressed these disguised transcripts directly from DNA molecules inside the lab cells to ensure the resulting RNA perfectly mimicked natural host transcripts.
And the second format?
Well, for the second format, they looked at the viral sequences in their natural, unadulterated state as replicons. These are basically just the self-replicating viral RNA sequences doing their normal thing, completely lacking those artificial human front and back covers.
And the data from this comparison was incredibly stark, wasn't it? When the viral sequences were dressed up like human transcripts, when they had that 5' cap and 3' poly-A tail, the host cell's rules suddenly applied. The poor codon usage severely reduced protein expression.
Yes, the virus was suddenly caught in the translation bottleneck. But when they looked at the replicon format, the virus in its natural state, the codon usage penalty completely vanished.
Wait, really? The non-optimal codons didn't slow down translation at all?
Not at all. It completely bypassed the penalty.
This is a massive revelation. It proves definitively that the non-optimal codons themselves weren't the secret weapon. The virus's immunity to the host rules wasn't in the sequence of the amino acids. It was a structural trick.
Exactly. And the researchers traced this structural evasion tactic to a specific region of the RNA strand, the 5' untranslated region, or the 5-foot UTR.
So the 5-foot UTR, that's basically the intro section, right?
Yeah, it's a fascinating piece of biology. It sits upstream of the actual protein building instructions. It doesn't code for anything. It's untranslated. Historically, a lot of people thought of these regions as just, biological filler or simple landing pads for ribosomes.
But the UT Southwestern team found that the 5-foot UTRs of incredibly diverse viruses all shared the seemingly magical ability to drive translation completely independently of the codon usage rules.
And the sheer diversity of the viruses they tested is what makes this a blockbuster paper. I mean, we were talking about Coxsackie virus, B3, SARS-CoV-2, hepatitis A, and hepatitis C.
Wow, those are all over the map.
Exactly. Evolutionarily speaking, these viruses are vastly distinct from one another. They occupy totally different branches of the viral family tree, yet they all possess 5-foot UTRs that function as this ultimate translation cheat code.
Okay, time for another quick bullet summary to lock this in. Bullet 1. Viral genes forced to look like human genes fail at translation. Bullet 2. Natural viral replicons completely bypass the host's rules.
And bullet 3. The evasion tactic is housed in the 5-foot UTR, that untranslated intro section of the RNA.
Love it. So think about this for a second. Think of the 5-foot UTR as a specialized lockpick. If you knew multiple distinct viruses all convergently evolved to use this exact same lockpick, what would that tell you about the host's lock?
Well, it tells you that the lock itself must be a fundamental universal choke point in human biology. If every successful thief is targeting the exact same wire in the security system, that wire must be the master switch.
Yes. Which brings us to the core biological breakthrough of this deep dive. What is that universal master switch? How does the host cell actually enforce its codon rules, and how does the 5-foot UTR break them?
To understand it, we actually have to completely discard how we usually visualize a strand of mRNA.
Right, because textbook diagrams have done us a bit of a disservice here.
Oh, massively. We typically imagine mRNA as a straight piece of string, a linear sequence of code being read from left to right. But in a normal, healthy human cell, mRNA does not stay straight. Remember that 5' cap and the 3' poly A tail we just talked about?
Yeah, the front and back covers.
Right. They aren't just decorative. They're highly active docking stations. Specific host binding proteins physically grab onto those ends. The eukaryotic translation initiation factor 4F or EIF4F binds to the 5' cap.
Okay, EIF4F is on the front cap.
Exactly. Meanwhile, the poly A binding protein, or PABP, binds to the 3' tail. And once they are anchored, these proteins have a strong affinity for each other. They physically link together, pulling the two ends of the mRNA strand with them.
Oh, wow. So they snap the ends of the string together to form a closed circular loop. Let's ground this with a visual memory trick. Picture a standard shoelace. Normally it just hangs perfectly straight, but the EIF4F and PABP proteins act exactly like your thumb and index finger. They grab the plastic tips at both ends of the shoelace, the aglets, and they pull them together, snapping them into a continuous circle.
That is a perfect analogy. And the researchers discovered that this circular closed-loop geometry is the absolute key to everything, because forming that closed loop creates a physical feedback mechanism for the ribosome.
Okay, walk us through the actual machinery of that translation.
Sure, so it happens in two main phases, initiation and elongation. Initiation is exactly what it sounds like, assembling the ribosome at the starting line. A pre-initiation complex, specifically a subunit called 43S, binds to the RNA to get things going.
So 43S is at the start line.
Right. Then there's elongation, which is the actual active process of reading the code. The full ribosome, made of the 40S and 60S subunits, moves down the track, reading the codons and chaining amino acids together.
Okay, let's picture this on our circular shoelace. It's basically a biological racetrack. Because the track is a closed loop, the end of the elongation phase is physically right next to the starting line of the initiation phase. This perfectly couples the two processes.
Yes, exactly.
So if the elongating ribosome, the 40S and 60S, hits a stretch of non-optimal codons, it has to stall out and wait for those rare delivery trucks to arrive. This creates a severe bottleneck, a massive molecular traffic jam.
And because the mRNA is physically circularized, that traffic jam in the elongation phase backs up right into the starting line. The slow elongation sends a physical or chemical suppression signal to the 43S initiation complex.
It essentially yells across the gap, "Stop initiating new translation, we are entirely backed up down here."
Precisely. The 43S complex shuts down. No new ribosomes are loaded onto the strand. The host cell's safety mechanism perfectly suppresses the production of proteins made with non-optimal code.
Okay, here's where it gets really interesting. Viruses do not play by these rules. Here is the physical mechanism of the viral sabotage. Viral RNAs either entirely lack that 5' cap and poly A tail, or, as the researchers discovered, their 5-foot UTRs are so highly structured, so densely folded into complex 3D shapes like tangles and pseudoknots, that they physically prevent the mRNA from forming that circular loop.
Yeah, the 5-ETR acts as a rigid, tangled spacer. It just refuses to bend. Because it prevents the binding proteins from connecting, the viral mRNA never snaps into a circle. It remains a straight, linear string.
And this is the brilliant, brutal simplicity of the viral cheat code. Because the string stays straight, the starting line and the finish line remain microscopic miles apart. When the elongating ribosome hits those non-optimal viral codons and causes a massive traffic jam further down the string,
The initiation complex sitting at the 5' end has absolutely no idea.
Exactly. The feedback loop is severed, the communication line is completely broken. That traffic jam cannot send its suppression signal back to the start.
So the 43S initiation complex just keeps firing. It keeps loading new ribosomes onto the strand, completely ignoring the host's codon usage control system, and blindly pumping out massive quantities of viral proteins.
It is just a stunning piece of microscopic structural sabotage.
It really is an elegant subversion of a deeply ingrained cellular rule.
Time for another recap. Bullet 1. Host mRNA forms a closed loop via caps, tails, and binding proteins, EIF4F and PABP. Bullet 2. The circular loop allows slow elongation traffic jams to shut down the initiation start line.
And bullet 3. Viral 5-occult UTRs physically block circularization, breaking that communication line and allowing rampant protein production.
So visualizing that biological racetrack, if you were designing a broad-spectrum antiviral drug right now, what geometric shape would you try to force the viral RNA into?
Well, you'd want to force it into a circle, right? You have to reconnect the start and the finish lines to reestablish the host's quality control. And incredibly, that is exactly what Liu and Liu's team did in the lab.
Oh, wow, they actually did that.
Yes. This is the smoking gun of the entire Nature paper. To validate their findings, they actively engineered circular mRNAs that contain those tangled viral 5-foot UTRs. They essentially used molecular staples to force the viral string into a closed loop, completely overpowering the 5-foot UTR roadblock.
That is amazing.
And the moment they forced the circle to close, the biological map instantly corrected itself. They completely restored strong codon usage dependence. Because the loop was closed, the traffic jam feedback signal was reestablished. The viral evasion tactic was defeated.
That is such a profoundly satisfying experimental validation. But let's connect this to the bigger picture, because why does this matter to you and me today outside of a specialized virology lab? This exact geometric dependency is the fundamental reason why the viral genes used in modern mRNA and DNA vaccines must be heavily codon optimized.
This is a massive real-world application. Think about the goal of an mRNA vaccine. To get the human body to accept the vaccine's instructions, say, to build a harmless viral spike protein so your immune system can recognize it, we have to package the vaccine in a format the human cell will accept. We have to give the vaccine mRNA a 5' cap and a poly A tail.
Wait, but if we give the vaccine transcript a cap and a tail, our cellular proteins, EIF4F and PDT, are going to grab those ends and snap the vaccine RNA into a circle.
Exactly. The vaccine mRNA is inherently subjected to host circularization. So if pharmaceutical scientists simply copied the natural viral genetic code, which is full of those rare, non-optimal codons, and dropped it into that circular delivery system, our cell's traffic jam mechanism would trigger immediately.
Right. The ribosomes would stall on the rare viral codons, the traffic jam would back up around the closed loop, and the suppression signal would completely shut down the vaccine's protein production before our immune system ever had a chance to build immunity.
That is precisely why vaccine developers spend massive amounts of time and computational power codon optimizing the sequence. They painstakingly go through the genetic code and swap out all the viral gargantuan codons for human huge codons. They have to replace the rare genetic dialect with optimal host sequences so that the ribosomes can speed around the circular track without jamming.
Mind-blowing. So what does this all mean for the future of biology? As we mentioned at the top of the deep dive, the editors at Nature highlighted that this paper does way more than just solve a viral mystery. It proves that mRNA circularization is the key determinant of codon usage effects in our own human host cell transcripts.
Right, and that is where the controversy and the excitement lie. For decades, the field viewed mRNA circularization as a sort of, structural quirk, a nice feature that perhaps aided in stability. This paper elevates circularization from a structural footnote to the fundamental physical governor of cellular translation.
It rewrites the textbooks, but of course, answering one massive question always opens the door to a dozen more. We now know definitively that breaking the circle breaks the feedback loop, but we still don't actually know the precise atomic level mechanism of how that slow elongation traffic jam physically sends the suppression signal to the initiation phase when the circle is closed.
This raises an important question. The physical proximity is there, but what is the actual conversation? Are the stalled elongating ribosomes physically bumping into the 43S complex and knocking it loose? Is the stall causing a shape change that ripples through the RNA strand? Or are they releasing a localized distress chemical? We just don't know yet.
And furthermore, we don't know exactly how many other human viruses rely on this exact 5-foot UTR structural trick. They tested a few incredibly diverse ones, which strongly implies a universal rule, but there is a whole vast universe of pathogens out there.
It's a phenomenal challenge for the next wave of structural biologists. Let me hit the final recap here. Bullet 1, forcing viral RNAs into a circle stops their evasion. Bullet 2, this geometric mechanism dictates how we must design effective RNA and DNA vaccines.
And bullet 3, the discovery entirely redefines how basic human cellular translation is governed. So, knowing that there are massive gaps remaining in how that feedback loop actually communicates, how would you design the next experiment to eavesdrop on that molecular conversation?
Yeah, that's the multi-million-dollar question. It will likely require next-generation cryo-electron microscopy to actually catch these molecules in the act of signaling.
It really is thrilling. We've gone on quite a journey today. We started with a seemingly dry mathematical paradox about codon usage bias and genetic dialects, and it unschooled into a high-stakes biological heist involving shifting geometries, structural roadblocks, and microscopic traffic jams.
And I want to leave you with a final thought to mull over as you process all this. We tend to think of biology as a chemical soup, just enzymes and proteins bumping into each other in a liquid. But if our human cells evolve this beautifully complex circular loop to carefully regulate protein production, and viruses convergently evolved rigid, complex, 5-foot UTR tangles specifically to break that geometry.
Wow, yeah.
What we're looking at isn't just a chemical battle. It is a billion-year-old architectural war.
An architectural war happening right now inside every single one of your cells. It makes you wonder what other geometric tricks, what other physical lockpicks are hidden inside you right now, just waiting for the right experiment to uncover them. Keep questioning the physical, microscopic world around you because, as we learned today, sometimes the most profound, world-changing discoveries are hiding right there in the untranslated margins. Thanks for joining us on this deep dive.